Greetings, Brookings Bio students. This is Mrs. Rydell, and in this slideshow, we're going to start our new chapter, Chapter 10, talking about cell growth and division. How is it that cells can go from a one-celled, you came from a one-celled little zygote cell to become a multicellular organism made out of billions of cells? And we're going to look at, kind of connects to some of the things we have learned in previous chapters. Remember in chapter 1, one of the characteristics of living things is to grow and develop. And in our cell parts chapter, we learned about some different cell parts like nucleus and cell membrane. And we're going to kind of revisit those as we go through and talk about how is it that cells divide. One of the reasons that cells divide is something called DNA overload. As the cell gets bigger and bigger and bigger, the demand on the DNA to tell all the different parts what to do becomes greater and greater. Remember, nucleus is the control center of our cell, and the DNA that's stored inside has the information in it to tell all the cell parts, other parts, what to do. It's coding for proteins that do things in the cell. And so as the cell gets bigger and bigger, and I get more and more cell parts, more and more cytoplasm, more Golgi, more endoplasmic reticulum, more ribosomes, it gets harder and harder for one nucleus to control all the things that are going on inside the cell. Think of it if you have a library with only so many books, and as your town grows bigger and more people use the library, it may be harder to get the book that you want right when you want it. You may have to go on a waiting list to read some of the more popular books. And think about how that works in your life. If you go to first hour class and you go to math and you get a math homework assignment, and then next hour you go to English and you get an English homework assignment, and then you go to biology and you get a homework assignment for biology, and pretty soon you start feeling pretty overloaded. It's like too much to do. And that's what happens in a cell. As a cell gets bigger and bigger, that one nucleus finds it harder and harder to do what it needs to do to control and provide the information for all the goings on that's happening inside the cell. One of the other reasons that a healthy cell might divide is that it has trouble exchanging materials with the outside. Remember, one of the characteristics of living things we learn in chapter 1 is living things obtain and use materials and energy. And so those materials, the oxygen, the food, have to be brought in across the cell membrane. That was our transport chapter. We learned about how things move in and out of cells. And as the cell gets bigger, the demand for transport gets bigger and bigger. If you look at these two little examples here, look at this little tiny cell. If I'm transporting oxygen in or food in across the cell membrane, I don't really have very far to go to get just about anywhere I want to be in that cell. But if I have a big cell, look at the distance I have to travel. If I'm moving something across the cell membrane, I have a lot farther distance to go to get around inside my cell. And that's a problem as the cells get bigger. It's harder and harder to get the materials that they need to keep the cell going. The ability to transport oxygen and food and waste across the cell membrane really depends on how much membrane you have. And that's surface area. Remember, the cell membrane goes around the surface of the cell. So if I look at surface area, how much surface I have, that's a indicator of how fast or how quickly I can transport things into my cell. The need for those things depends on the volume. The stuff that's inside the cell needs more oxygen for all the mitochondria. I need to get in glucose. I need to move ions around. And if you look, as cells get bigger and bigger, those two things, surface area and volume, don't increase at the same rate. If you look at a cell, and we're going to use some examples of some different sizes of cells. So if I could imagine a cell as kind of a cube shape, and notice this cell is a 1 by 1 by 1 centimeters on each side. It's a 1 centimeter cube. This cell is a little bit bigger, a 2 by 2 by 2 centimeter cube. And this cell, even bigger, a 3 by 3 by 3. So let's compare the surface area and the volume on these cells as they grow. Okay, if I'm growing from a small cell to a bigger cell, we could use some math to figure out the surface area of an object. And remember from math, length times width would give you surface area of this side. And if I have a cube, I have a side here, and I have a side on the back, and I have 4 sides around here. I have 6 sides on my cube. So if I did the surface area for one side and then multiplied by 6, I could do some math to figure out what the surface area of my cube is. And I could do that for all 3 cubes. Then if I look at volume, now remember volume is length times width times height. If I calculate the volume of my cube, right, I could do that for all 3 cubes. And then let's compare those two. If I do surface area to volume, make a ratio, what I see is that in a very small cube, it has a pretty high surface area to volume ratio, 6:1. But look at as my cell gets bigger and bigger and bigger, the surface area doesn't grow as quickly as the volume. So in a 2x2x2 cube, my surface to volume ratio is only 3:1. It's cut in half. And look at a 3x3x3 cube. Surface area to volume is only 2:1. And that difference in the surface area and volume ratio makes it so it's harder for cells to transport the things that they need as the cell gets bigger and bigger. Bigger cells need more food, they need more oxygen. And as they reach a size where they can't transport fast enough, the cell's going to get a signal that says, hey, it's time to divide. Because one of the ways that I can take a big cell and make a better surface area to volume ratio is to divide the cell in half and make a smaller cell. It's the way that multicellular organisms grow, by increasing, remember we talked about increasing cell number. I can grow a little bit bigger, but once I reach a point, I can't get any bigger. You're not just one big giant cell inside. You're made of lots and lots of little cells, and it makes it so they can function and do their jobs to transport the things that they need quick enough. Okay, let's dust off our brain and think back to some previous chapters, because remember, biology is one of those subjects where you can't just do a been there, done that, forget that stuff now. Because things come back from previous chapters to the new chapters, and it helps to connect everything together and make things make more sense. So we had these 2 vocab words before. DNA can exist in different forms in the cell. As unwound a little bit, it's still coiled, but not as tightly wound. Spread out, we said spread out in the nucleus of a non-dividing cell. We called it chromatin. Chromatin is the loosely coiled form. And if I want to move my DNA around, a lot easier to move it around if I scrunch it up, and if it's in the scrunched up form, we call it chromosomes. And the cell can switch back and forth between those 2 forms, depending on what's happening in the cell. If I'm doing my job and wanting to know the information about how to do that, the DNA has to open up so I can read. It's kind of like reading the book. You have to open the book and look to see the pages. If I want to move my DNA around, the cell is dividing, it's a lot easier to move it around if it's scrunched and packed tightly. Think about when you do your homework. When you go home from school, you pack all your books and your homework into your backpack because it's easier to transport that way. But once you get home and you want to read the information, you have to take the books out of the backpack, open them up so that you can see the information and know what to do. And the cell does that with its DNA, switches back and forth between those 2 forms. Let's remind our brain a little bit about how DNA is different in prokaryotes and eukaryotes. Remember when we did our cell parts chapter and we colored our cell picture with all the parts, we had a little picture up in the corner of a prokaryotic cell. DNA in a prokaryote, it has only 1 chromosome, and it's circular. It's in a loop form. And remember, prokaryotes don't have a nucleus. So DNA is in the cell, but there's no nuclear membrane around to protect it. And actually, bacteria actually kind of tack their little loop of DNA attached to the cell membrane to keep it from kind of floating away. Eukaryote DNA is a little bit different. We have our DNA in rod shapes, linear chromosomes. If you take up 1 chromosome and stretch it all the way out, it's 1 long piece of DNA. And it's not in a loop form. It's linear. And many chromosomes. You have 23 pairs of chromosomes in each of your body cells. And remember, eukaryotes, their DNA is found in the nucleus. They have a nuclear envelope around their DNA. It includes plants and animals. Fungi have a nucleus. Protists, little pond critters, have a nucleus. Anything that's not a bacterial cell has their DNA enclosed in the nuclear envelope. Okay, let's get some new vocab now, because when we talk about chromosomes, we need to have some names to talk about the different parts. And by the time I can see a chromosome, when the cell is dividing and the DNA has packed tightly, usually when we look at a chromosome under the microscope, we see it in this form as kind of an X shape. This is one part of the chromosome, this is the other part. And those two arms that make up the chromosome are called the chromatids. So this is one chromatid, and this is an exact identical copy. It's like a Xerox machine making a copy of the information in this part of the chromosome. And those two copies are held together by something called a centromere. Centromere is the area in the middle that holds the two chromatid arms together. If you look inside your body cells, you have two of every kind of chromosome. One came from mom, one came from dad. And so inside eukaryotic cells, we have what we call homologous pairs or homologous partners, because all of our chromosomes come in pairs. So this would be an example of homologous partners. Notice now we got some vocab words that sound a lot alike, very close. Chromosome, chromatin, chromatids. You got to be careful on the vocab words this chapter, because we have a lot of vocab and a lot of words that kind of come from similar root words. If you look most of the time though in our cells, the chromosomes don't exist with copies attached to them. We don't really see them in that form. This is the kind of spread out, use the information form. So if this is one homologous chromosome pair, one from mom, one from dad, and notice the little bands here stand for some different genes. Homologous partners, if you squish a cell and look at the chromosomes, you can match up the ones that are the same size and same shape, and they carry genes for the same trait, like eye color genes or hair color genes or height, or all of the information that tells you how to be you. They are carrying the same genes, but they don't have to have the same gene choices. Homologous chromosomes are not exactly identical. Remember, chromatid arms on chromosomes are identical, but homologous partners don't have to be exactly alike. They carry genes for the same traits, like eye color genes, but they don't have to have the same choices. Dad could give you a blue eye gene, mom could give you a brown eye gene. If you look at cell division now, because bacteria cells have such a simple DNA setup, one loop chromosome, their reproduction is a lot easier and simpler than eukaryotic. They don't do mitosis like we do. They use a process called binary fission. Binary fission. Bi means two. Fission means to split or break apart. So we're breaking or splitting our cell into two cells. And basically all bacteria have to do is make a copy of their little loop of DNA. And remember, it's attached to the cell membrane, the plasma membrane. And then all I have to do is grow a little bit, stretch my membrane apart and pinch in the middle, and I get two cells. Bacteria can do that pretty quickly. A bacterial cell can reproduce in about 20 minutes. And it's because it only has one chromosome. It doesn't have to have a big complicated line it up in the middle, hook on the spindle fibers, make sure everybody's going where they need to go, because they just have that one chromosome. Remember, we saw that word back in our cell parts chapter, and we talked about evidence for the endosymbiotic theory that bacteria and chloroplasts were once prokaryotic ancestors that were taken in by a cell and not digested and stayed to live symbiotically in the cell and become the mitochondria and chloroplasts over time. One of the evidence for that is that mitochondria and chloroplasts reproduce using binary fission, just like a bacteria cell does. So let's vocab. Cell cycle. Cell cycle is the series of events that cells go through as they grow and develop. And so think about it's a cycle because it can repeat over and over and over again. We're going to watch a little animation here. And if you look at the cell cycle now, cells can go through, let's get rid of that little noise. Maybe we need it for volume. Starting with G1, there's a series of steps. Copy my DNA, make cell parts for the new cell, go through the division phases, mitosis, and then it can go back into the cycle and go again. Cells can repeat over and over and over, and once they divide, they can divide another time. And so that cycle, that process, the series of steps that cells go through as they go through their life is called the cell cycle. Okay, let's go back to our slideshow. And so what we're going to learn in this chapter is those steps. What is it? What are the steps that cells go through when they undergo cell division? And so we're going to go through it quickly and talk about what happens in each phase, and then we're going to color a little picture. So interphase is the non-dividing part of the cell cycle. Cells are not dividing. In G1, they're growing, they're hanging out in the body, they're doing their job being a nerve cell or a blood cell or a muscle cell. And because we need to know the information about what to do, the DNA is spread out as chromatin in interphase. Once the cell gets a signal, for whatever reason, to divide, it's going to copy its DNA, make all the proteins that go along with it. Remember, proteins that help to wrap the DNA up and into chromosomes. And then it's going to make all the other organelles and molecules needed for cell division, make some new mitochondria and more Golgi bodies and all those cell parts that we need for the new cell. And that's going to also cause the cell to grow a little bit bigger. And then we're going to go through the division phase. And I know you remember this from 7th grade. You had a chapter on mitosis. Dividing up the nucleus, the chromosomes, prophase, metaphase, anaphase, telophase, and then splitting our cytoplasm. Each new cell needs to get some of those cell parts, and that's cytokinesis. And then we have a little phase where the cell actually kind of leaves the cycle and stops dividing, and it's called G0. So if you look at a cell, at the cycle now, remember it's a repeating cycle. And some cells go through the cycle very quickly, and some cells not so quickly. And so we're going to make a little picture here of what's happening in the different phases. So I'm going to end my show here, and I'm going to pull this picture up. You should have this now. And what we're going to do is color a little bit. And I'm going to ask you to pick, you can pick any color that you like, but I'm going to have you save red for later. So pick another color that's not red, and we're going to do a little coloring here. And we're going to start with G1, because that's kind of the phase most cells are in most of the time is interphase here. So we're going to color in our G1 phase. And then I'm going to write some stuff down about what happens in this phase. If you're coloring with a really dark color, kind of save a little space or a little room around your labels so that you can tell that this is the G1 phase of the cycle. And so let's write some stuff now about what's happening in G1. G1 is the G for grow. We have some growth happening in G1. Originally, when scientists named these phases, they named it gap, G for gap, because when they looked at cells under the microscope, they could see some things happening in the S phase, and they could see some things happening in the mitosis phase. They could see the chromosomes, but it looked like in these other two G phases that they didn't really see very much happening. But now we have better instruments. We know a lot of chemistry is going on. So gap has been changed to growth, G for growth. This is the phase of the cell cycle where the cell is doing its job. Hanging out in the body, being a body cell, a nerve cell, a muscle cell, a bone cell. and basically it's being a body cell in your body. And cells spend most of their time in this phase. Cells spend most time here. And now if I'm going to be a body cell, I need to know the information about what I'm doing. And so if you think about that, remember DNA forms chromatin, chromosomes. In this part of the cycle, DNA is going to be spread out in the nucleus, unwound a little bit as chromatin. Because I need to open up the DNA so I can have the information tell me what to do when I'm doing my job. Now, signal comes to tell our cell to divide. And that signal could be I'm getting too big for my britches. DNA overload is happening in my cell. I can't transport nutrients fast enough. It could be an injury that happens. I fell off my skateboard and scraped my knee or broke my leg. Anything that tells the cell it's time to divide. And so this signal comes here. And once I decide that I'm going to divide, I'm going to make a copy of my DNA, because the new cell, when I split, has to have the information of what to do. So I'm going to color my next little section here. Pick a new color. And we're going to color S phase. S for synthesis. Remember synthesis, like protein synthesis? We talked about that word before. Synthesis means to build or make something. And what we're making in this phase is DNA. We're going to copy our DNA. And then once I have a copy of my DNA, I also need to make some new cell parts for the rest of my cell. If I'm going to make a new cell, I need to have some more mitochondria and endoplasmic reticulum and Golgi bodies and ribosomes and all those cell parts that we learned about. So in this phase, second G phase, G for not gap now, but growth, we're going to make all the other cell parts and molecules that we need for the new cell. Put my color here. Make molecules and cell parts for the new cell. Molecules and cell parts. And as I'm making more mitochondria and more endoplasmic reticulum and more Golgi bodies and more, it's going to make my cell grow bigger. This is also a growth phase. And if I'm going to be using my DNA information to tell me how to do that, how to make more molecules, I need to have my DNA opened up as chromatin. Notice in interphase, these are all interphase, parts of interphase, kind of like little subphases. I'm going to put a little arrow here to help me remember that this is a cycle. So starting with G1, I'm going to go to S phase, and then G2. Notice now, I haven't really divided anything yet. I've been getting ready, I've been making the things I need to divide, but the cell hasn't divided. This is interphase, the non-dividing part of the cell cycle. And it's made up of these three phases, G1, S, and G2. They're part of interphase. Okay, now I'm ready. I made a copy of my DNA and I got all my cell parts ready to go. And now I can actually do mitosis. I'm going to do the dividing part of the cell cycle. I'm going to get some colors here. And mitosis. Now we're going to go through those phases that you remember from middle school. Prophase, metaphase, anaphase, telophase. And I'm going to put a little dividing symbol here, like a math symbol, to remind me of what's happening in this part of the cycle. Mitosis is the cell division. We're going to actually divide the cell. And notice prophase, metaphase, anaphase. These phases, what we're dividing is the nucleus. We're going to divide up the DNA. divvy up the chromosomes into each of the two daughter cells. In this part of my cell cycle, I'm going to divide the cytoplasm. All of those cell parts get divided up. So my new cell has all the cell parts of mitochondria and Golgi bodies and endoplasmic reticulum and lysosomes that it needs. And so if I'm going to move my DNA around now, DNA in this part of my cycle is in the chromosome form. And that's the actual cell division part. Notice cells don't spend very much time in mitosis. Majority of time is in interphase. We have one last little thing to add to our picture. And that's why I said save your red for later. Because if you look at the top here now, this is like a little detour out of the cell cycle, where cells actually leave the cycle and stop dividing. And notice it looks like those letters look like the word go, G-O up here, but this is not O, this is 0, this is G0. And really what it means is not go, it means stop. I'm going to make a little stop sign here. S-T-O-P, because in this part of the cycle, the cells actually stop dividing. And they hang out, they're still being a body cell and doing their thing, but they don't go back into the cycle and divide again. An example would be a nerve cell. Remember, once nerve cells, think about nerves in the spinal cord. If they're injured in a car accident or a diving accident, those cells don't divide and replace themselves like a broken bone or a skin knee. And so we say they're in G0. And think about if we could figure out the mechanism that causes cells to go from a non-dividing back into the cycle and divide again, we could get people who have spinal cord injuries and are paralyzed to repair those injuries and be able to walk again. And scientists are working on studying cell cycle and studying how to do that. They've actually taken stem cells in mice with damaged nerve cords and injected stem cells and actually gotten some growth back on those spinal cord nerves, which isn't really going back into the cycle and making the nerve cells divide, but it's a way to try and fix this. So as you look at the cycle now, some cells go through the cycle very quickly. Every second you're making a couple million blood cells. Bone marrow cells divide all the time. You're always making new blood cells because they get kind of worn out. White blood cells get worn out and beat up fighting germs, and your red blood cells get kind of knocked around, squeezing through your capillaries, dropping off oxygen. So we're always making the bone marrow stem cells are always going through the cycle and making and dividing and making new blood cells. Some cells divide very rarely, like for example a liver cell may not divide very, very often at all unless you do something to stimulate the cell division, and like remove part of your liver to donate to someone who's having a liver transplant, and then those liver cells will grow back again, regenerate, and the piece of liver that gets donated to the sick person also will grow bigger once it gets in the new body. So that kind of takes us through the cell cycle, and we've gotten down our information about all the phases. Thanks for watching.